not seeing anything. I will call him. Once. I'm in the right meeting. YMS BKSU SWG. Wait. Deepak. Very wrong time. Hi Himanshu.
Harriet.
Uh, who let you in actually? Uh, in this, do you know?
I joined through the Google Calendar,
Oh, I see.
and there was I joined automatically.
1 minute. Sachin, दीपक को कॉल वो देना फोन. I can't see any invitations here. Like people are trying to join, but I can't see anything. Like I can't let them in. Yeah, so people are waiting, but I can't see them. So I have asked Deepak to join and like add them. Just waiting for them to like being added, and then I will start. Deepak, Deepak.
Hi, Atharva. Uh,
Hi Himanshu, tell me.
so you sent a Python notebook that you're going to go over today.
Yes, yes. One
Uh,
minute.
is it in the Slack
Okay,
channel?
वन मिनट वन मिनट मैं बोल रहा था दीपक है क्या पास में? चल ठीक है ठीक है ठीक है, he joined. Okay. Yes Himanshu.
Yeah. So where have you shared the notebook? It's not in the mail.
Yeah. So I have not shared the notebook because it's in Jupyter, and it will require some dependencies to be installed. So for today, what I will do is I will just go through it on video, and we can I will seek your inputs, uh, like what to change, what not to change, and we'll do it together
Okay.
basically today. Hi everyone. Sorry for starting late. There was some technical glitch. Uh, I will uh, start soon now. Uh, yeah. Okay. Deepak has added me as co-host. Now I can do things here. I hope you can hear me and uh, yeah, see my presentation also. So what we'll do, I will there might be some new people here, so I will just take 5-10 minutes to reiterate what we saw yesterday, and then start with some new stuff and then go to analysis part. So uh, what we saw that fiber photometry is an invasive technique in which we implant an optical fiber inside the brain of an animal in the region of our interest, and shine light through the optical fiber and record the out incoming light again uh, through some kind of photo receiver device to measure the brain activity in the region of interest where we have implanted. So I will 1 minute. I will just choose my pen. Okay, now I can see. So see, we shine light from here and then it goes to some input. So this is how we do it. In this we saw that this utilizes the uh, property of a genetically modified uh, uh, fluorescent indicators. So this uh, GEFI, genetically encoded fluorescent indicators, these are the, expressed in the brain deliberately by using some viral vectors, and these have a property that whatever uh, uh, analyte you want to measure, so you can measure dopamine, you can measure voltage, you can measure calcium signal. So basically you can design this indicator uh, to function in such a way that in presence of the analyte it fluoresces more. Basically it gives out more light when you shine particular frequencies uh, on that protein. So this is the property that it uses, and what we saw last time that uh, we use special properties of this indicator as a control, and that property is that if you shine some other wavelength, uh, and this wavelength is very specific wavelength, this can't be just any other wavelength. So this wavelength has a property that the emission from the JFE now are same in presence and absence of analyte. So any change now is like independent of the concentration of what you want to measure, or the state of the system. Basically if you are measuring voltage, it's not concentration, like it's a voltage or a voltage around the indicators. So this is what we saw, and what I do in lab is to measure dopamine concentration. So we will go through data that of uh, dopamine today. But before that, I'll just familiarize you with what we measure and how do we measure a little bit. So what we are shining is the excitation wavelength. This wavelength is isospastic, and this what we measure is emission wavelength. So this is emission wavelength. How do we measure? The hardware looks like this. There is some light source which shine excitation light or the isospastic wavelength through some optics in the brain of the animal. We collect back emission light which passes through a light detector where we measure the intensity of the light, and then we process and analyze the data. And we can do time synchronization with the behavior boxes from here. I will show you with the help of actual data that how does this work. Now how do we achieve like how does it look physically? This is just a schematic. How does it look physically? So for driving the light into the brain, what we need is of course LEDs, right? So these are the LEDs of different wavelengths that come in. So these have a property that they can shine specific wavelengths of light, and the intensity of that light from here can be modulated with help of current. So you provide a very precise value of current, and the light that is being shown from here, let's say of 465 nanometer, and from here is 405 nanometer, which can be an isospastic wavelength. So these LEDs are governed by a very precise LED driver. So this LED driver provides a very precise value of currents to LED, which in turn give out light. So this light now passes through an optical system which consists of some dichroic mirrors. So what do these mirror do is they allow particular wavelengths to pass through them and particular wavelengths to get reflected out of them. So this provides optical paths for different wavelengths, basically different optical paths for different wavelengths. So let's say you are shining the excitation wavelength from here, and so it will pass right through it and right through this dichroic mirror all the way, and through some connectors it will go to the animal's head. So these are all connectors, different kind of connectors, and similarly this wavelength 405 again, it reflects from this dichroic mirror and goes into the brain. What our goal here is to collect the same wavelength of light, 525 nanometer, that is like green light, from this system, and then sending it to some fluorescence detector. Now what is this fluorescence detector, we will see. And all of these can be connected to one integrated electronics system here, in which it measures the timing. So this LED driver needs a very, they generate current, right? So they have to generate current at very precise times that we will see just after 2 slides, that why do we need those precise timings. So this system here on top, this system can record these timings, the values of the current provided, and it also gets integrated with the output of the system. So this particular system in our lab, we use custom built electronic system on just a PCB. It's a Arduino based system. So this is just a data integrator. You can make it using any electronic device. You can even have a PC with microcontroller attached to it. So these systems use LED driver, LEDs, the optic system to separate wavelengths, then some connectors to the animal head, and back from animal head to fluorescence detector. So these 4 or 5 systems totally make the photometry setup. Now from this what we get is the fluorescence reading. So in the end, what are we recording? What we are recording, the intensity of emission coming out from the brain. So this emission is from a specific wavelength. In our case, it can be 510 nanometers. So what are the units of this measurement? What are we recording in? So we are recording in arbitrary units basically. So it depends on the detector. So in case that we use a photodiode, so what does a photodiode do is the photodiode converts incident light into current. So if you shine some light, it will give a proportional amount of current out of it. So now you can measure the total amount of light that is being shown to on the photodiode and it converts into current, and that can be digitized and recorded by your system. But you can use other kind of detectors, camera or a photomultiplier tube. Camera is also quite prevalent, but it is more noisy. But you can get a 2-dimensional data out of it. So basically, if you are recording from multiple fibers at once, let's say 10-20 fibers, then you can record using camera each of them as an image. But with photodiode, what it does, it sums all of them up in one singular value. So there are different kind of detectors you can use, but in the end what we are interested in delta F and not the arbitrary value of F, which is fluorescence. So units don't matter here that much. What we call it as the output as fluorescence. So we will call it as F from now on. So what are the possible sources present in this fluorescence signal? One of the obvious one is fluorescence from the indicators that comes out like the actual signal as well as the like the baseline fluorescence out of the indicators. Then there is this concept called autofluorescence. So this is a property of materials to fluoresce in light. So any material can fluoresce theoretically. Like you shine light and it will emit some other wavelength, some larger wavelength of light. So you can get autofluorescence from patch cord or other optical components. You can get autofluorescence from the brain tissue also. So here on the right side, this is an example of mouse intestine under different wavelengths of light. So this is with no special proteins basically. There are no light dependent proteins here. This is just how intestine looks under different kinds of light. So if you get movements in the tissue itself, for example, some blood vessel might fluoresce, and the blood flow may change your fluorescence output. So this is one kind of noise that we don't want in the signal. So there are ways to reduce this autofluorescence from patch cord. Like there are special materials out of which this patch cord means the optical fiber. I forgot to mention. So patch cord is the optical fiber that goes from the optical device to rat's brain. So there are different kind of material, like companies manufacture them, and also there are maintenance tips for them. By that you can reduce these values. but this is what you can like, this is dependent on the sensor property and the tissue property. You can't do much about it. And then there is a bleed through of excitation light or background illumination. So this is again dependent on the optics inside your setup. And the background illumination is like, if you have many connectors in between, right? So from optical device to brain, you have different types of connectors to connect different patch cord to the brain. So the light can leak through it, so the ambient light can also go through it, and you see a very clear spike when you turn on the light or turn off the light. That's why what we do is we do our experiments in red light. So red light doesn't fall in our recording or excitation spectra. That's why we do it in red light. But you can also choose to do it in white light. There are ways you can find out that signal after collecting the data. We'll go through it. Now, how to find out, like, so for this specific kinds of noises we use, and especially like movement. So movement may change all of these. So any artifacts because of these sources can be found out by using an isoasbestic signal, basically another wavelength that we saw, which is not dependent on the analyte that we want to measure. But how to shine both the lights at same time? We can't do that, right? If you shine both the lights at same time, you get a summed up emission, and you can't measure that. So you have to do it at separate time. So if you are measuring out the same emission wavelength, you have to do it at separate time. So what we use in our lab, there are again these timelines can vary, but what we do in our lab is so this is on x-axis there is time, and in this time is in microseconds. So what happens here is that first of all we do baseline measurement. So we measure the intensity of emission wavelength that is coming to the system without shining any light, without shining isoasbestic, without shining excitation. That gives us a baseline correction value, and then what we do is we turn on the isoasbestic LED here, and this LED may have its own kinetics of turning on, right? So as we give the signal, it may have the actual LED turning on is this. So this needs to be calibrated before you set up the system. This you should know these values, like after giving current, how much time does the LED take to come to peak power, and also the biosensor might have its own kinetics. So once you shine LED, after how much time you expect that fluorescence. So depending on that, you give this gap. So this gap, after this gap we do emission measurement. So this measurement, like you take many readings in this, and then average it out, basically over some 1 millisecond or so, you average that out. And then what you do is you again give some gap to the system, and then again do baseline measurement here, and turn on the excitation LED and measure emission here, this. So what you get is this is an emission measurement, this one first and this one second. So these are around separated around 4 to 5 milliseconds in time, and this can vary depending on what system you choose, so or what kind of sensor you have, but these are considered as coincident measurement. So any change in this value here if it correlates with this value here, that means that it is not due to whatever analyte you wanted to measure. So this is how you counter, how do you get two measurements at near at very near time, in very near in time, so that you can control for noise or artifacts. So now after this, how does this data look like? So this data look like this. So you have a data type which is an intensity of emission wavelength after you shine excitation light, intensity of emission wavelength when after you shine isostatic light, and there is this time column in microseconds. So in our system, like, it was 0 microsecond and 5000 microseconds, so basically around 5 millisecond difference. And then you have the flow intensity measure in arbitrary units, and now you have basically a time series for both types of data. One is actual relevant data, and one is control data. So you have to use this control data to reduce artifacts from this. There is this third type of data, which is basically the integration of behavior to this system. So this we can study later on also while discussing code. But what it does is, so our photometry system works on a clock of a particular computer or particular system. Our behavioral events happen in some other so this photometry system does not know about what behavior is going on, so you have to convey that information somehow. So we give these kinds of messages to this system, so that the time is synchronized in both. So we know that behavioral event 3 happened at 0 microseconds in this particular clock. Another event happened at 20,000 microseconds. So this will now this is basically synchronized data. So how do we pre-process these data now? So that's what is the main topic. There is someone in chat. What are the data types here? So I don't know, what do you mean by data types? Like if you can space like
Yeah, hi. So yeah. So I'm asking about the data types written in the very first column, say E, I, and somewhere it is MSG.
yeah. So this is just my notation to do it. Like you can have different data types also. If you follow some libraries to analyze, they have their own standardized data formats. But these contain all the information that you have in any kind of data format basically. So you need a time and you need a fluorescence signal. This is a tabular format. You can write it in CSV file, you can write it in binary file, whatever file you can choose, and then read using any notebook, any software basically. You can write your own Python notebook or any library that can read that data. So I save it in CSV files basically. So the data will be saved as E, 0, 170.43, then E, like that. So
That's okay, but my query is regarding this. I mean, what does this small e signifies here? Small e and
so
then
E
R.
is basically uh, this value of emission. So you measure in one cycle, you measure emission twice. One for isostatic excitation and one for like your main excitation wavelength. So isostatic excitation, this one acts as a control which does not depend on the analyte you want to measure. Basically, in my case, like, it doesn't depend on the dopamine present in the region, but this signal is expected to rise with dopamine concentration. That's what. So this acts as a control and this acts as a main signal. So what the main pre-processing steps are listed here. So this graph may seem quite busy, but it's not. So the data looks something like this. So on this column, middle column, the data is presented on on 4000 second scale. The same data is now zoomed to a 60-second scale, and you see here in the third column. So what you do is first you do a filtering step. Basically, you don't get data without noise. So there are all different kinds of noise, like electrical noise, uh, or some low frequency noise based on the like ambient light can also be present. So you have to remove that, and after removing you get something like this. The data, the signal decays in time. So this decay is due to a property called bleaching. So this bleaching property is a property of fluorescent indicators, any fluorescent material actually in biology, uh, where the light can cause chemical changes in the fluorescent material and render it permanently uh, disabled basically. So it does not fluoresce after uh, uh, it has some property of being inactivated permanently. So if you keep a fluorescent molecule in under light for a long enough time, it will become inactivated after some time. So uh, some proportion of the molecules get inactivated during what we measure. So you get this kind of slope out of your signal, and this slope is seen in both the signals basically. So because the number of fluorophores now are decreasing, you see a decrease in both the signals. So for a long time I was also under assumption that since number of fluorophores decrease, the slope will be similar for both isoasbestic and the main signal that you get. But this is not in the like I always get it like that, but this is not always the case. You may get different slope, so you have to correct accordingly in later step. So if the purple one is your control signal, then you have to fit that control signal onto the main signal, and that fitting may reveal some kind of so you can correct for bleaching using that fitting. You can also correct for this kind of motion artifact. So you see these increase. This increase is also present here, which is not zoomed in. If you zoom in, then you will see. So I will show you that data today. I will show you how does it look. Then you see that what percentage of this main signal, the green one, is explained by the purple signal. So are there changes in the green signal which are also present in purple signal, and you subtract those changes. Once you subtract, you get something like this, and you divide by the mean value. We will see these steps in detail now. Okay, so I will move to my notebook. So this here you get the final signal. You can choose to convert it to z-score, and we'll see why is that important in later. So I'll go to my notebook. So this is a Jupyter Lab notebook, and I will require your inputs here while I go through it, and I will ask you questions like what happens when we change some parameters. So these are all dependencies that I will install. This is just the format of output I want and format of plots I want. I am reading my data files here that can be
Increase the font size a bit. Font size.
okay. I'll increase that. Font size. Okay, I'll zoom in like this. Also, I will request people to speak out if they have doubt, because Deepak might not be around. So I will not be able to see messages that often. So if you have any doubt, just speak up whenever you want. Barnopriyo has one doubt. Do we need to assess goodness of fit during the second step? And I will come to it and explain then. Like we'll discuss this then. So this is how my message file look like. Messages, time, and the event type. There are many events that happen in my behavior. And this particular event type 1 here, in my case, it signifies the my session start, basically the time when the relevant behavior starts and ends in my setup. So this says that I started recording 4 minutes before the session started, and my session lasted for 30 minutes. So here you already see that ideally this should have been exactly 30 minutes, 34.299, but there is already some 10-15 milliseconds difference between the photometry clock and my behavior systems clock. So that's why I don't take only one initial signal, but I take many behavioral events. Like all the behavioral events I transfer to this photometry system using this time synchronization step. Basically, I not only send the session start and session end readings, but I also send many like when the lights were turned on, when the speaker was turned on. Though all those signal goes to my photometry system, and that's why I can synchronize time live. I don't depend on one clock forever. So what I will do here, I will assign this as my session start time, and once I have done that, I will align all my message times to this particular session start time. So I already did that. This is very specific to my notebook. And so what we see is session start time in photometry file is now 4 minutes. So 4 minutes after starting recording, I have started my session. And in my case, the behavioral event of most significance is the lever press. So now I print this just for a sanity check that the first press was recorded 0.2 minutes after session start and last press 25 minutes after session start. So this is very specific. You don't need to see this. There can be different data types also. So there are different systems which I have used. One of them is manufactured by a company called RWD. So it gives out this data in different formats. So you can read that format by some other code, but that's not the main point here. So the main point here starts with this. I read the fluorescence data raw. So this looks like isoasbestic and excitation combined, alternating signals I get, and the corresponding F value I have. So I can do any condition. Basically, I want only the fluorescence. In this condition, basically I will apply that the time should be positive. Basically, positive time implies that during my session, any time before the session, I have already set it to negative time. So either this condition or a general condition which does not apply any filter, I can choose anything. So for now, let's say I do not apply any condition, and then what I am doing here is just cleaning the data and making different data sets. So now I have divided it into FFE and FFI, just like I have showed. I have made two tables, with one of them containing excitation data, other one containing isoasbestic data. So now we have exactly what we wanted. So now starts the main pre-processing step. I will plot this. So this is how the data looks like. So you see here, this green one is excitations and orange one is isoasbestic. So this is how the signal looks like. And you see these kinds of changes, these abrupt changes here at before 0, before the session starts. So these kinds of changes are the ones that you want to find a control for. And what you can do, so this may happen because probably there was light turned on before this time, the ambient light, and then I closed doors of the setup, and so that both the signals got down together. So it is generally recommended in every library that you will find that you need to see these kinds of artifacts manually, and you need to figure out that does the data look good or not in general. Do you find these kind of abrupt changes within the session? And you have to do some manual checks basically on the broader scale as well as minor scales also. So for example, this kind of dips you see here, if I zoom in to some level, you see these dips are correlated somehow. So this kind of wavy pattern is seen in both my control signal as well as the excitation signal. I can zoom in more. So if you see this, I can zoom in more on basically 1 second scale. I will show you some readings here. You will find it interesting that in the raw data also you can see some kinds of readings. So these are my behavioral events. You see that after red and green line you find a peak kind of thing here, here. Here. So these, so you can basically zoom in and out of the signal and start analyzing it right away, like from this particular, like just visualize this. So this is very important step to do it. Now I will go back to home. I will apply a filter where I ditch this data because this data will affect my calculations later on. So I will just apply this filter again. I'll go up. And I have applied a filter where it will take only the time after during the session and not before the session. So with this trick I get rid of any data. Now the data size will be smaller, and I will plot it again. So now you see that it starts from the session and I have gotten rid of that particular set. So I don't want this, I just close it. Now you see these kinds of this particular kind of noise is very sharp noise, and these are typically the kinds of, when this is, you see this is only present in one of the channels. Now you will say this is only present in excitation channel and not the isoasbestic or our control. So this might be related to dopamine, but it's not the case. These kinds of very sharp, this is actually a single data point if you see. So these kinds of sharp noises are generally due to electrical noise in the surrounding, and this may affect one particular pin or any port in your system. So you have to remove this independently in both the signals. So at this time I will read some doubts. What here leak time set input in 56. Okay, so this was the step that I don't want you to focus on. Basically, these are just how I process my uh, behavioral events. So my behavioral events are recorded in uh, microseconds. So I'm converting everything in milliseconds, and that's why, or even in seconds. Sorry, sometimes in seconds and sometimes microseconds. So this is just a pre-processing step I do. So lick times is how may like when the rat touches the lick sensor, but it's not the relevant topic for now. Uh, is here isoasbestic signal the control signal? Yes, yes, that is the control signal. So yeah, so for removing these kinds of sharp noises, what we use is a filter called median filter. So this is the median filter, medfilt. It is in a library called SciPy. So what it does is, uh, this median filter is applied on a signal uh, with a particular kernel size. So what it does is, if you have kernel size of 5, it will take 5 consecutive readings and take the median of it as the signal at that particular time. So it will take t minus 2, t minus 1, t0, t1 and t2 signals. Basically, t minus 2 is 2 time steps back, what did you get? 1 time step back, what did you get? t0 is current time step, and then further 2 time steps. This is for window size 5. Similarly, you can have different window size. So with of these 5 values, you find median and report that as your signal. So you can have different kernel size depending on your data, how much data you want to average. That on the basis of that you can get this median filter. So this is generally robust to these very sharp noises of 1 or 2 samples. So 1 or 2 samples don't affect median that much, and we know that in general from our experience we don't get it more than 2 or 3 samples, these kinds of noise, consecutive samples. So I apply it as kernel size 5. So if I apply kernel size 5, this noise goes away, the sharp noise that we see here, but not other noises which occur at least for 2 samples, they will not go away. So if I apply even kernel size 3, you will see it goes away because it occurs only for one particular sample. So that one sample will not affect median of 3. But 2 samples, if the noise occur for at least 2 samples, then it will affect the median of 3. So that if you know that your noise occur for more than 2 samples, then you have to choose a kernel size of at least 5. That's how you choose this kernel size here. And if you see the signal side by side, so this is the raw signal, this is a denoise signal, and the green lines that you are seeing here, the green and red, are my behavioral events basically. So just don't focus on them, the signal is important. So you see the reduced width of isoasbestic signal here, the reduced amplitude of isoasbestic signal. That may be because of my access also, but this reduces some noise overall in the system. So the second type of filter is called a low pass filter. So this low pass filter, okay, so what we do with the low pass filter is that, as the name suggests, low pass, it only allows low frequencies to pass through the signal. So any high frequency noise, so if you zoom in here, you see many these kinds of fluctuations you don't expect in biology to occur at 1 millisecond scale with, in particular with dopamine. So you want to smooth these uh, traces basically. So you apply a low pass filter which can take care of high frequencies, uh, high frequency noises. For that you need a parameter called sampling rate, like at what rate you are taking samples. So what sampling rate means, that values collected in 1 second for one channel. So here if you see, I will tell you this is my excitation signal. I am taking the time column of that signal and taking the difference. So it is giving me at every 74441 microseconds I take a reading for excitation signal, which comes out to be like 7.4 millisecond. So for 7.4 milliseconds in 1000 milliseconds, the frequency comes out to be 134. So I take 134 points in 1000 milliseconds. That means that it is 134 hertz. So again, this parameter depends on what sampling rate you are taking, and this sampling rate again depends on your LEDs, ramp time, your biosensor, what are the kinetics of the biosensor. So this sampling rate depends on the plot that I showed you, how are we managing our excitation and isospastic sampling. And then I give it a Butterworth filter here, which I apply like low pass. These are the parameters for Butterworth filter. These kind of details, like what goes in this Butterworth filter, we will study when we are going to the math concepts of low pass filter. So that time we'll study like what these parameters mean, but basically this is not allowing any frequencies more than 30 hertz to go into it. And this is a function that applies that. So just note that if you are not aware of low pass filter, just write this term down, that I need to understand low pass filter when we are teaching maths, and what does this butter or filter mean. So that when we are teaching math, we'll go through this in detail. And so if you apply this Butterworth filter, then you get this kind of reduction in high frequency noise here. I'll show you here. So this is how isospastic signal looks like from 1493 to 1495. Just you see the changes now are more smoothen in the right one, in which we have applied the low pass filter. So in this it was very noisy, but now it is not, because we know that in our biology system such kind of noise don't exist. Anyway, so now this is an important step that I mentioned about photo bleaching, right? That this signals, they deteriorate, not deteriorate, they decrease over time, like this here. And we need to take care of that, because what we assume that any change in this signals when analyte is present happens proportionally to the signal. So if you have a photo bleaching like this, then use pen. If you have a photo bleaching like this, and you have peaks in the signal, which are like actually signal, which are not noisy peaks, which are actually peaks, that will look like this here. So let's say dopamine comes here and your peak looks like this. Here the peak will be proportionally small. So this peak height in the first case will be higher, this will be lower, even if the same concentration of dopamine is released. So what you need to do is you need to take care of this photo bleaching to get the real signal out of it. So what you do is you apply now a high pass filter first. So that high pass filter, what it does is that it flattens the signal. So currently if you see, it is again photo bleached signal, it is decreasing over time by around 10 arbitrary units. It is decreasing, like from 175 to 165. This high pass signal, what it does is, high pass filter, it just take it to 0. So if I want to explain you in simple term, for any signal, uh, the mean of the signal is a 0 frequency term. So basically, if you apply Fourier transform on it, like what Deepak taught in last lectures, if you convert this into time, from this time to frequency domain, if you convert this signal, then the mean of the signal will come at 0 frequency basically, and around that the other frequencies will be added up. It may be little hard to understand if I am not showing any plots or something, but we'll cover it in signal processing section. How does this high pass filter work? But so basically, these slow noises which change the mean of the signal are removed, and this flattens the signal to a mean value of 0. So now basically the signal has become is centered around 0 for overall time. And now you see these dips and these kind of features. So these are still common in excitation and isospastic. We have not corrected for that. Like, we have not used control till now. We have just processed them together. We have not used control to actually refine our excitation signal. So what do we do here? From any guesses from here? Like, how do we use this control signal to remove noise from excitation? Like artifacts, not noise. Remove artifacts from our excitation. Any guesses, so that I know you are still here?
Well, the signal shows sort of very high growth or sort of increase or decrease compared to sort of average, and plus you can do sort of a standard deviation of all the trials. So what I'm saying is there is just too much change in signal, that particular thing can be removed,
No, but
because that might be some
we
artifact
have to
or
use control signal. So what you are saying is remove too much changes and all. That we have already done using median filter, the low pass filter and high pass filter. But what I want to say, that we have not used our control signal. So this particular orange plot, that is our control. So these all changes are due to some other artifacts which are not dopamine. But I am interested only in dopamine signal. So I want to see what changes are present in my excitation signal, but not in isospastic signal. That's my goal, right?
just subtract the isobastic signal.
So how do I do that? So you might have used this, not in this context, but like very extensively, the method that when there are two time series signals, you get let's say temperature and humidity. You can explain humidity by temperature. You can explain temperature by humidity. Like, what the term is called is correlation, right? So these are correlated signals. So these changes are common to both excitation and isospastic. So what we find, what we do is we regress both excitation against isospastic. So regression means that what changes in excitation are being explained by isospastic signal. What So you basically try to estimate excitation signal using isospastic signal. This even can be done. Basically, if let's say temperature humidity are goes in perfectly parallel, like as temperature increase, humidity also increase, then you can estimate temperature very precisely by humidity, and there are no other factors affecting temperature. You will know that. But let's say there is some other factor affecting temperature, and you want to find its effect on temperature. Then what you will do is you will regress temperature with humidity. You will estimate that, and you get let's say you get an estimate of T cap. So humidity gives you value of T cap, and the actual reading is T. So whatever is the difference now, T minus T cap, is the actual effect of the other variable on that. So the same thing is here. So you take excitation minus excitation. Let's say our excitation signal is called F. So you take F minus F cap, where F cap is the signal that you estimate using isospastic, and F minus F cap is now your signal that you assume that it is coming from dopamine. You can safely assume that it is coming from dopamine because you only see it in excitation wavelength and not in isospastic signal. So that's what we do in the next step. Basically, linear regression of excitation by isospastic for motion correction. So this term is called motion correction because we attribute these kinds of changes, these kinds of rough changes, to movement of animal, of the optical elements in the of the patch cords and all in the setup. So if you have not understood, I can try to write also. Yeah. So basically, if this is your fluorescence signal is Fe, and other is Fi. Fi is your control signal. This is your excitation signal, which is the signal of relevance. Now what you do is Fe estimate. You find an estimate for excitation using a simple linear regression. Basically M into Fi plus C, where M and C are obtained using simple. So this is what you do, and now what you do, the motion corrected signal is equals to Fe minus Fe estimate. So this is the summary of the next step that we will do. I hope you get it. So this is just how you do the linear regression here. You take high pass filtered isospastic signal, high pass filtered excitation signal, then you regress them and you do y minus y prediction as FFE motion corrected. So this is how you get motion corrected signal. I will show you that. So this was my high pass signal at 656 here, let's say. Yeah. So these were the kind of changes that you were seeing before motion correction. So focus on the green one, that is like our excitation signal. Now if I zoom in here, those signals should go away. So these kind of changes that you were seeing, this particular increase, this basically low frequency kind of wave. That wave is somewhat still there, but more or less gone. So some of them might be actually the true signal that may still be present, but it's the pattern is not there anymore. So if you want, I can also plot this one. Oh yeah, so yes, what I missed here is this was the pattern that we saw. So using that we predicted, sorry, I presented it mistakenly. So this was the prediction, basically the brownish signal you saw is the prediction that isoasbestic does. So these kinds of changes are basically artifacts, you don't want that. So you subtract this and then get this particular high pass signal. So now if you plot
Hi, Atharva.
yes,
Hello.
yes,
I have a doubt. Why do we actually regress it, like predict and then subtract rather than directly subtracting the signal? Because isospecific signal would
I see.
basically mean, I mean, any changes in isospecific signal would be like, in my head, and I am wrong, is the fact that there would be this isospecific signal is could be directly subtracted from the emission signal, right?
Yeah, yeah.
But why do we do this prediction?
So yes. So what happens that you can do this. That's what I was saying in between, and I stopped. That this linear regression step can also be done at this stage, where you have not applied the high pass filter, but you have just the denoise signals, which have you applied a low pass filter and the met filter. So you have these kind of bleaching still there. You can apply linear regression here, and you can't directly subtract because the scales may be different. So here you are looking at like both are very same, but the mean values are different. So if you record at some other intensity of isoasbestic light, this orange signal may be like at 50 arbitrary units, or it may be at 200 units. So you have to consider that what linear regression basically is doing is that bringing it to same scale, and then we are subtracting. So it is multiplying every orange point to bring it here, bring it at the center of green, and then we are subtracting. That's why we do linear regression. But after applying high pass filter, it will not make a change. Like high pass filter already scales them to same mean, so it will not make that that much a difference. It will be the exact same thing. Please. Okay. So now what we have in our hand is high pass motion corrected excitation signal. Yes. So now what we do? So as I mentioned, where here. Oh, actually the thing is gone. So I mentioned that the peaks at these points may be larger than peaks at this point, because they scale proportionally to the signal. So what we need to do is divide them by the signal value, so that we can get a proportional change in the f. So what you do is after high pass filter, what you get is del f. Del f is like f value subtracted by the total mean. So del f divided by the f value at that particular time will give you the final signal. So you need to find the f that you want to take. So f is changing, you can't just take anything. So what you do is you fit exponential to these signals. So I will show you how we do that. done. So to get the mean like what to divide with the f value, you fit an exponential curve here. Now Barnopriya was asking that do we need to assess the goodness of fit during the second step? Yes. This is can be done manually by seeing like what kind of fit you are doing. Generally it is assumed or by like the empirical evidence of these fluorophores that they the bleaching happens at a double exponential rate. So we fit a double exponential to them. This is a double exponential. Basically it's a form of a to power b to power x. Anyway, you can do this fit. Yeah, you can speak, Banu.
No, I think I might have misconveyed. I mean, yeah, fit in this step as well. But what I was asking specifically was, do we need to check the goodness of fit when you fit isosbestic to excitation?
Oh yeah, this one, like when you fit. So
Yeah,
what's the regression
exactly.
coefficient? Correct. So yeah, one of the introductory paper on photometry does this, and uh, like in the absence of movement, like when you are, let's say, doing it in an artifact free environment, basically in a very controlled environment, let's say in a petri dish, if you do this, then you should not get any motion artifacts or in the signal. So basically you don't want to see this kind of positive correlation there in an artifact free environment. But in our case, since there are movement present, you can see a positive correlation in the signal, because the movement, if it affects one of them positively, it should affect the other one also positively, and this correlation basically we try to minimize by subtracting the f excitation minus f excitation estimate. So goodness of fit, like we want least fit possible in a noise free environment, like artifact free environment. It will be a very bad fit. Any fit is like movement. So I don't know if it answers your question.
Yeah, it kind of does. What I was thinking was like, at some point, obviously, like, do I reject my regression and try again or something like that? Like when I'm fitting this step. So I was wondering what is the way to go? Like, what do I look for? But I get you.
Yeah, so I can tell you where it fails. So let's say if this green signal, which is our excitation signal, and the control signal, if they bleach at different rate and you apply direct linear regression, then it will fail, because both of them have different slope if you apply at this particular step, if you don't filter that. So then linear regression will give you a bad result. So then that time that's why you need to observe these plots and get double exponential fit of them and check if they are going through the same rate. So you can have some kind of metrics for these values, like um, are they going at the same rate? Shall I do high pass filtering before regressing, or after like after high pass filtering I should regress? So that kind of checks can be done and should be done. Yes, if you mean goodness of fit, like.
Yeah, thank you. Yeah.
Okay, so now what we have is we have a high pass motion corrected signal which is del f. Basically on y axis it should be del f, and on we have an exponential fit of for the f. So this is again not exact, so people have different ways of doing this. You can also take local mean of some values as your mean signal, but I didn't find them very accurate in my case. So there are different ways to do it. I am just mentioning that this is not the only way, but it works for me. So you take an exponential fit, this will give you the f value, this will give you the del f value. What you do is now this del f you divide by f value and multiply by 100 to get a percentage df by f. This is percent uh, percentage df by f value. And you can also z-score this value uh, to uh, uh, so yeah, to you z scores you can compare across sessions, like in say across sessions and animals you can compare. But again, people have raised questions on this, that how does it fare across sessions and across animals. Even I am not very well versed with that, so there are different papers on it. I will just show one of that here, like this: a statistical framework for analysis of trial level temporal dynamics. Like how does it fare in across sessions, across animals, across time in sessions. So there are different ways, very highly mathy ways. So I have to also read it, and if I eventually learn this, I will like to go through this also in the end of this course. But anyway, for now, we assume that these z scores can be compared across sessions. And if we plot this, you can get something like this. This is our final signal, and these blue lines are and red lines are my behavioral events. I will show some of these. Let's say this particular portion. So if you zoom in, you see some peaks around here, around the blue lines, and by seeing, like when I saw these data for the first time, I was seeing nothing basically. Like I was pretty surprised, there are so many peaks. How should I like, it may be just be a random chance that these peaks are occurring after blue lines or red lines. Yes, that can be the case. So what you need to do is now collect all these. Basically you collect one red line that signifies one particular behavior event, let's say press, and you collect 500 milliseconds before, 500 milliseconds after, for all such events, and then average them together. So that I do it in a different notebook. So that notebook basically makes the trial averages. It's not very important to see how I process that data, but what is important is you see these. Where is it? Oops. There is an error. 0 to yeah. So this is one of the last plots that I'll show. It's not loading or. So eventually what you do is you align the plots and get some readings out of them. Okay, it's not working. I will show you one. So this example will give you that what kind of things we can get. Oh, it's taking time to load. That's it. It was already there. So many plots are open. Okay, it's not opening. I will go through the summary from this. So what we did, even overall, that we had a green excitation signal, which is our main. Then there are these control signals which contain motion artifacts generally. So we do some filtering and all for removing electrical noise, and also we do low pass filtering to remove high frequency noise. High frequency noise can be generated by many electrical noise present around the area, and within system noise. So they use 30 hertz cutoff. I used 30. They use 10 hertz cutoff. I used 30 hertz cutoff. Then what we do is we fit an exponential. We also do bleaching correction by basically by dividing the signal with this exponential fit. And what we also did was they did bleaching correction before doing the regression, and they did bleaching correction using an exponential. What I did was bleaching correction using high pass filter. So both are acceptable base. What you want to do is just remove this uh, like trend, basically decreasing trend in the signal, and then you regress both of them together, basically try to estimate the green signal using the control, and that is called movement correction step. And after doing that, you get a movement corrected signal, and then you normalize it by dividing it with the value that you get here, this f. And you can take normalize value, and you can also take z-score out of it. So that gives you signal for both. You can also do the same step on isospastic, and you see these kinds of signals you get in the end. Like if these are your behavioral events, the blue ticks, then you get like peaks around that in only in the excitation, but not in isospastic. So that is a control that these peaks are really associated with the behavioral event and not like a fluke. Anyway, the plot is not working somehow. I hope my if this would have worked, like it would have been nice. Oh, I can show you my already. So just one last plot I wanted to show. Yeah. So this is kind of stress that we get of dopamine around my press time. So you see these peaks on an average, not before the press, not very long after press, but somewhere some time after the press. And you see these peaks only in the rewarded case, but not in the unrewarded case. And this is an evidence, like when I measure dopamine in that particular region, you get a very nice increase in dopamine after reward, but not in the case of not rewarded cases. So these kinds of analysis will yield interesting insights on how neurotransmitters behave in behavior. Especially research in case of neuromodulators like dopamine or norepinephrine, and these are quite frequently studied using fiber photometry, and new very good sensors have been developed for studying these neurotransmitters. There is a bundle fiber photometry also, which I may introduce later. That using that you can study cellular level details. And calcium imaging can be done like this to see the broader activity of neurons, not neurotransmitters. Field of dopamine has benefited a lot using this technique. So you may read, just if you are interested, like photometry and dopamine, you will get many results out of it. So this was my today's part. I hope you got the summary. I request you to note down any mathematical terms that you didn't get and send to us on Slack or anything. Like, we will try to cover it in our next lectures. Anyway, we have planned it, but if you request this particular term, I want to know how, or this particular step, how do you decide parameters or something? How does the met field work? How does field filter work? Then we can go through how does this curve fitting, goodness of fit, what do you mean by that, those terms, like those kinds of question. If you request us, then we'll cover those specifically. and this will help you in building this. So this is a custom built pipeline. These same steps are available in different libraries. Like you don't need to go through them like math of each one, but just apply one library, give it the data, and it will give you final output. So, but it should not be a black box for you. That's why it is I suggest that you build your own notebooks at, to start. So yeah, that's it. Do we have any analog like? Oh, welcome, Bhanupriyo. Uh, Spike sorting from data from bundle photometry. So, spike sorting in IFIS, to those who are not aware, is based on the concept that the same spike is being recorded by different tetrodes or different electrodes. Like same signal is being recorded at different spatial locations, same signal. But in fiber photometry, it's not the case. In bundle photometry, you assume that one signal is only in one particular fiber. They are spaced apart like that. I think so. I have not read extensively on that. I had initial plans to do that, but now like what we get is you can put a camera and image the bundle basically using the camera. So you get pixel. Each bundle has like pixels, some pixels. So uh, uh, you know you may get correlations between bundle of fibers, but how do you deal with that? I don't know. I'm not really sure. Just my guess. Is there no case of dip in baseline dopamine when there is no reward? I think I read somewhere. Okay. Okay. I will have to go back a lot. Means that is a very specific question, which is okay. My AWT slips. Yeah. So in this particular animal, in that region of recording, we get a dip. In that animal, we didn't get. So, and see these are the kinds of signal that you get. So this is actually, you see a small shading around the plot. That is like the ACM or standard error of mean, the variance around that line, and the signal is very clean. So you can really get very good signals out of these sensors if used properly. Okay, so that's it for today, I guess. If no one has any doubt, welcome. I will end the meeting now. Thanks for attending, and if you have any doubt, please share on the Slack.